I'm Alison Pettit. I'm Director of Biomedical Research for MARTA Research, and I'm our representative on the TRI Research Translational Committee that is responsible for creating and organising this new seminar series that we've started that is really trying to shine a light on successful research partnerships that have actually facilitated and driven translation of research into clinical outcomes. So it's my pleasure today to introduce Associate Professor Kristen Radford and her research partner, Associate Professor Mireille Lahoud, who's from Monash University, and they will be telling us about a really wonderful story looking at the development of vaccines for cancers. Thanks, Kristen.
Thank you. Thanks for having us. I'm going to hand straight over to Mireille. What we thought we would do is tell you just the pathway of how it started. And I'm more at the translational end and Mireille's more at the basic science end. So we're going to start with her story first. What do we do? Do you share your screen, Mireille? Is that how it works?
I think so. Let's try.
Okay, go for it.
So this is a project or collaboration that started over 10 years ago between Kristen and I now, with the aim of translating dendritic cell biology into next generation vaccines. So this year or 4 years, I don't have to tell you what a burden of disease infectious diseases are and why enhancing immunity through vaccines is critical. Similarly, enhancing immunity through cancer immunotherapies offers us new ways to offer new therapeutics. And the way we like to address this is using dendritic cells. Now, why dendritic cells? Dendritic cells are professional antigen-presenting cells. Their role is to pick up antigen, process it, and present it to T cells to initiate CD4 and CD8 T cell responses. As such, they're critical for initiating and maintaining immune responses. Also, as such, we can use these to manipulate immune responses, both in a prophylactic and in a therapeutic setting. Our preferred approach for doing this is to use antibodies to dendritic cell receptors and attach antigens on the end. That way, when you inject these constructs into the body, they home directly to the dendritic cells and are specifically taken up by the dendritic cells in order to mount immune responses. Sort of like addressing an envelope rather than posting it publicly on Facebook. This approach was first pioneered by Ralph Steinman, and he applied this approach for delivery of HIV and tumor antigens to DEC-205, considered the gold standard in the field. But the story was a little more complicated than this, in that dendritic cells are heterogeneous. There's not one dendritic cell subtype, but multiple different rare dendritic cell subtypes that have some common features, but also are tailored for unique functions. And the cell types that I was particularly interested in is what we now call the conventional DC1 subtypes. These are the mouse CD8 or CD103 DCs. In humans, they have CD141. And they're particularly good at cross-presentation. What this means is they can take up exogenous antigens, so material from dead and dying cells, such as infected cells or tumor cells, and present it on MHC class 1 to initiate killer CD8 T cell responses. And that's where my interest in dendritic cell biology started. What are the receptors that enable the CDC1s to survey their environment, take up antigens, process them, etc. What can this teach us about dendritic cell function and receptor biology? But importantly, how can we apply this towards immune modulation? And today I'd like to briefly tell you about one of our receptors, which is CLEC9A. CLEC9A is a C-type lectin-like receptor that we discovered on mouse and human DCs. It has a single C-type lectin-like domain, extracellular C-type lectin-like domain, and a cytoplasmic hemitamp signaling motif. It's conserved between mouse and humans with 53% amino acid identity, but importantly, every ligand or interacting protein we have found to date is cross-reactive between mouse and human CLEC9A and interacting partners. So it has a species-conserved function. It's selectively expressed by these cross-presenting DCs in mice, humans, and most mammalian species. And it recognizes dead cells. It's an endocytic receptor. It assists in dead cell uptake, but it's not critical for dead cell uptake. What it is absolutely critical for, though, is the cross-presentation of dead cell-associated antigens. So you need CLEC9A in order to shuttle the material that you acquire from dead cells into the right pathways in order to initiate CD8 T cell responses. We identified its ligand as F-actin. Now, actin is known as a universal contaminant, but why would you want actin as a ligand? You know, in hindsight, it actually makes a lot of sense. Actin filaments are evolutionarily conserved across most species. They're cytoskeletal components, they're only revealed upon cell death. But importantly, they're never found on their own. They're always coated in actin binding proteins, both from the host, but also any pathogen that infects cells tends to use the actin cytoskeleton either to get into the cell, to replicate, or to get out of the cell. So if you were to sample the actin of a cell, not only do you know that the cell has died, but you know what the cell was doing at the time of its death. So you can consider it the flight box recorder of the dead cells. Or you can think of it as acting like a fishing net. When CELAC9A samples actin, not only is it sampling the actin itself as a fishing net, but it's also putting in all the associated proteins such as viral or tumor proteins that were associated with that actin. So CELAC9A actin recognition enabled dendritic cell uptake and processing of antigens that were associated with dead cell cytoskeletal proteins for cross-presentation. We generated antibodies to CELAC9A. Initially to mouse CELAC9A, we put antigens on the end, and we measured the responses either to these antigens or initially even to the rat backbone as a surrogate antigen. What we found was if you immunize mice with CELAC9A, with antibodies to CELAC9A, you can induce antigen-specific immune responses that are considerably higher than other receptors. So here we're looking at targeting CELAC9A compared to other receptors on cross-presenting DCs, including CELAC12A and DEC205. You can see these are very modest in the absence of adjuvant. In fact, both CELAC12A and DEC205 need adjuvant to get a even vaguely useful response, whereas CELAC9A does that without adjuvant, although you can boost it further with adjuvant. We also generated antibodies to human CELAC9A, and we found similar to mouse CELAC9A, antibodies to human CELAC9A were able to induce potent responses in the macaques. And if we added adjuvant, these responses were maintained or slightly elevated. So we identified that the anti-CELAC9A antibodies were particularly good at inducing antibody responses, both in mice and non-human primates, even in the absence of adjuvant. But if you want to induce cytotoxic T lymphocytes, you absolutely do require adjuvant. Can we apply this to vaccine development? We started off with an influenza M2E peptide. This is highly conserved among flu A viruses, but it is very poorly immunogenic. So this is just a short sequence. On its own, it's poorly immunogenic, but we tried adding it to the end of the CELAC9A antibodies. In this case, we put 3 copies in tandem. We found a single injection of CELAC9A coupled to M2E, provided long-lived immune responses that went over 1.5 years, provided disease protection 90% of mice, and if you gave a boost, you could have 100% disease protection. Now, how do we apply this to human translation? I'm going to hand over to Kristen. And now you're on big screen.
Thanks.
And I'll hand over to you.
So I do this. Where's the share screen thing gone then?
I'm going to go back to Zoom. So I think.
There we go. Right. Share. Okay. So that was a lovely introduction and over, 10 years work. And where we were at the time was messing about with dendritic cells and using them as vaccines. And the way we were doing this was just taking the naturally circulating dendritic cells from blood, which are less than 1%, enriching those, activating them and administering them back as a vaccine. And lots of other people were doing this at the time as well, using monocyte-derived dendritic cells. We knew this was a fairly safe and effective approach, but one of the big problems was that this beautiful CELAC9A antibodies and potential there, we didn't even know what the human equivalent cells were that they were targeting. So that was a big problem. And what we started doing was starting to dissect these different dendritic cells in humans. At the time, none of the markers that were used to segregate mouse and DCs actually lined up. So it was a real challenge back in those days. And to cut a long story short, we we're using this marker called CD141 to identify this really rare subset of dendritic cells that we demonstrated functionally had very, very similar properties to the DC ones in the mice. And so using this functional analysis and also were able to demonstrate that they exclusively expressed CLEC9A in humans. This was a really seminal paper that allowed us to align mouse and human DC subsets and translate a lot of that mouse knowledge on the DC ones across to human systems. What it also enabled, these are really, really rare. We needed a leukapheresis product to get a couple 100,000 cells. So it wasn't very easy to be able to validate any human-specific targets of these molecules because they're just so rare and hard to find. And so all the work that we've done over the last 10 years or so has really started to develop novel ways of isolating these cells, of growing them. We've got our humanised mouse models, etc., that we can use. And so this just then ties back into Mireille's story of we thought these DCs were the bee's knees and decided that we needed to harness those for immunotherapy. She had a molecule that could do just the job, and that was really the beginning of a 10-year plus collaboration now to try and translate this knowledge into the clinic. It was really able to be kicked off by this wonderful PhD student at the time, Kirstine Tullett. So she spent half of her PhD done with Mireille generating a series of human antibodies and fusing various antigens onto the end of them. And then she spent the other half of her PhD up here using our human models to be able to validate those. And that was initially funded. We seed funding from Mater Foundation and the Prostate Cancer Foundation of Australia. And that enabled us to get some bigger fish, including a nearly $1 million US Department of Defence grant and another one from Worldwide Cancer Research in the UK. And so where we've been over the last 10 years is together we've designed a bunch of human constructs that we could use to evaluate whether these vaccines would actually be useful in humans. We've got two different tumour antigens that are widely applicable across a huge range of different cancer types. But the one we started off first was with the CMV PP65 protein. And we also, for benchmarking, because the DEC205 antibodies were already in clinical trials, we wanted to benchmark against that. And we also used an irrelevant control, which was in essence just soluble protein conjugated to a beta-galactosidase antibody. So I'm not going to talk a lot about all the data that we've done because it has been 10 years of blood, sweat and tears, literally. But these antibodies really retain their specificity for that human DC1 subset. And a key difference between mouse and humans is that CLEC9A does bind to plasmacytoid DCs in the mouse, but doesn't in the humans. So that's already a key difference. This is just some of our data from the PP65 story that showed, and we're actually really pleased but somewhat surprised, that the CLEC9A targeting antibody is far superior at delivering these antigens to activate CD8 T cells compared to DEC205 and compared to targeting other DC subsets. We're also quite surprised because the paradigm would suggest that the DC2 subset is better at inducing CD4 T cell responses, but our CLEC9A antibody delivered to DC1s again was really, really good at activating CD4 T cells as well. And it was at least as good as targeting to other subsets or using DEC205 antibody in that regard. Moving on to our tumour antigens, we've now been able to show these are 4 different tumour epitopes, 2 against the NY-ESO tumour antigen and 2 against the WT1. And you don't need to look too closely in any of the data, but what you can see is that our CLEC9A antibody activates these CD8 T cells across all 4 of these different tumour antigens across different HLA alleles and 2 different antigens. And they're better than untargeted protein, and in most cases, except for this one, they're better than the DEC205 as well. So that's really exciting and encouraging. The other thing that we've been able to show is just using patient samples, and these are melanoma patients that had previously received an NY-ESO protein vaccine, and so they were known to have NY-ESO specific responses. We could just take their PBMCs and culture those with those antibodies. And again, you could see that the CLEC9A antibodies that we've made are far superior at reactivating T-cell responses in those melanoma patients compared to any of the other benchmarked vaccines. And this just shows you that they also have enhanced killing capacity against tumours that express their NY-ESO and the correct MHC. So one of the other things that's really helped translate this is the humanised mice that we've developed. These are mice in which we engraft human CD34 progenitor cells, and they go on to develop a human immune system. And we've shown over many years that these are really great models for developing human dendritic cells and studying those subsets. And now we've more recently engineered these so that we can look at T-cell responses as well. And so we've applied these humanised mouse models to look to see if we can get priming in vivo of naive human T-cells. And with our CLEC9A antibodies, indeed we can. So if we vaccinate these mice with FLT3 ligand to boost the DCs, add in our CLEC9A targeting antibody plus poly-IC as the adjuvant, you can see that our CLEC9A antibodies are superior at priming those naive T-cell responses compared to the DEC205 or the untargeted protein. So that's a very short snippet of more than 10 years work and the models that we've been able to develop to facilitate that moving forward. So I'm going to hand back over to Mireille now, and then we can talk about where we go from here. Back to you. Mireille, you're on mute. Can you hear us? You're on mute.
We are now unmuted.
Lovely.
Can you see my slides?
Not yet.
Okay, let's try again.
Yep.
So where are we looking to go from here? We feel that the best place to start in terms of cancer immunotherapy is in the setting of minimum residual disease with a high risk of relapse. So right now we're exploring collaborations for lymphoma, AML, and ovarian cancers. We validated the CLEC9A platform in mice, humanized mice, and non-human primates now. So our next stages are further preclinical studies to produce lead human vaccines, evaluate their safety, immunogenicity, and efficacy in humanized mouse models with Kristen. And then we're looking to develop this by developing the lead vaccines and evaluate safety and immunogenicity in phase 1 clinical trials. This would give us a humanized CLEC9A tumor antigen vaccine for clinical development, and we've already been working on NY-ESO1 and WT1, as Kristen has raised. But also this will give us a validated platform for the next generation of cancer immunotherapy vaccines going forward for personalized medicines. Now, how does our partnership work? What's enabled it to work is we've had a shared vision. We both believe dendritic cells are the coolest cells in the immune system and in the body. So our vision is to understand both dendritic cell biology and to translate this into health benefits. We've brought complementary expertise. My expertise in mouse DCs, receptor biology, molecular immunology, and antibody engineering, and Kristen's expertise in human DCs, cellular immunology, cancer immunotherapy, and the awesome humanized mouse models. We started off with seed funding and a joint student initially funded by PCFA. This enabled us to generate the prelim data and publications that enabled us to get the big international grants, including the DoD and the Worldwide Cancer Research. And we bring together different strong collaborative networks. Kristen, do you want to add to that?
We've just about got it all summarised up. Is that pretty much reflected in what I would say as well? I think that's about all we've got to add, but we're happy to take any questions or any discussion on that from here?
Moving forward, we're looking for funding for the next phases. We're looking to establish stronger clinical and commercial collaborations, but we're looking forward to a COVID-free world where we can actually have our students and staff cross over between labs as we used to, because that was the fastest way to ensure training of the next generation across both molecular and cellular, mouse and human DC biology. And a note to thank our teams at both ends and our funding bodies, including PCFA, NHMRC, DoD, Worldwide Cancer Research, and Mater Foundation. And back over to Kristen.
Back to you.
Anyone have any questions for Kristen and Mireille?
Thank you. Great talks. And obviously, you're targeting it systemically. Is there any option to go skin-derived or mucosal dendritic cells? And I'm thinking, since we have access on level 4, you might actually have on your fingertips a commercial partner. I mean, obviously, they're putting dried protein on their velcro path, and it seems to work very well.
We've just...
Can you just check that Mireille heard the question?
Did you hear that question, Mireille?
No, not really, it was breaking up, I'm sorry.
So the question was regarding, could we be targeting skin or mucosal DCs and taking advantage of companies such as Vaxxus that have the nanopatch technology for delivery of this antibody. Do you want to answer that, or would you like me to start?
Do you want to start?
So I think from the mouse work, the vaccination works in any route. And we've just co-published a commentary editorial article about this and how we would move that forward. And that was one of the things that we raised in there. So there's been... I'll send it to you. So one of the things with that, there has been, and this has all been mouse, there was a little bit done in humans, is that the CLEC9A antigen looks like it could be down-regulated or more weakly expressed in the skin DCs. But even after injection into the skin, these antibodies, because they're small, they're going to traffic straight to the lymph nodes, and that's probably where you want them to be going. So they don't necessarily need to be picked up by the tissue antigen DCs, but as long as they can get to the lymph nodes, that's where they're likely to be most effective. At least that's what the mouse data would suggest to you. And following on from that, then it doesn't really matter where you deliver them. So the skin patch technology is something that would be really interesting to look at. Mireille, do you want to add to that?
No, I think that covers it. One of the complications for people working with human skin, though, is that what you need to break down skin to get the DCs out is actually fairly harsh treatment with different enzymes. And CLEC9A tends to be a little bit fragile on the surface of a cell, so it's been hard to tell in humans exactly how well expressed it is on skin DCs. But as Kristen said, it gets through to the lymph nodes regardless.
Anthony?
I saw a paper recently where they were showing that CLEC9A in human tissue expressed on macrophages, if I remember correctly. Have you seen that?
No, I haven't. Have you seen that, Mireille? So it's a question from Ranjeni about CLEC9A being expressed in macrophages in humans.
Hi, Ranjeni. Good to hear your voice. I have seen that paper. I have not seen any evidence of CLEC9A on the macrophages, so I'm not sure whether it was how the cells or staining was performed or anything else. We will look in more detail, but we've certainly not seen any evidence of CLEC9A on macrophages so far, neither in mouse nor in human.
I guess the other question that Ali might want to add to that is whether the macrophages have eaten the DCs or whether the DCs have got pieces of macrophages stuck to them.
So, which could come very much down to the way the tissue's been prepared, which is what some of our data is showing that we'll hopefully have published very soon. So
I need to read that paper.
Any other questions?
Yeah, I just have a one that you'd be expecting, but you mentioned clinical collaborators. So what does he plan? Are you going to target, or what's your plan to try and get clinical collaboration into this setup?
So we've been talking to a bunch of clinicians and also potential commercial partners as well. So I think it's going to come down to, we're keeping our options open pretty much at this stage, and seeing who's most willing to buy it. I think where the DC therapies have been most successful has been in the setting of AML, for example, where you get a setting of minimal residual disease, but high risk of relapse and no alternative treatments. And so, and ovarian cancer is another similar disease. They're both able to be targeted by both of our antibodies, actually. So you could even potentially do one and then the other to broaden the reach of the different antigens, of which we've got clinical collaborators around town and both here and down in Monash. So that's our target, and we'd like to be able to do that multi-site and to keep the collaborations across the borders and potentially internationally if we can. So that's where we are at the minute.
And the MARTA clinical trials unit has now got a phase 1 clinical trial capability. So that's an opportunity to leverage off that to run the first phase 1 clinical trials.
Yeah. Did you want to add to that, Murray?
I think you've covered it.
Any other questions from the audience?
Do you expect the antibody to be immunogenic?
So the question was, Murray, do we expect it to be immunogenic in human? Possibly not. So everything that we have done so far, it doesn't directly activate the DCs themselves. And if we just add them in, it doesn't indirectly activate T cells or anything. And the 2 papers that we put out last year, the reviewers really hammered at us on that. And so we made sure that we did it and did it well. So I don't think so. I think you're going to need adjuvant. Do you want to add to that, Murray?
No, I agree. Going for CD8 responses, we certainly need adjuvant. We've now done mice, non-human primates, and humanized mice and human DCs, and doesn't seem to be immunogenic in its own right. But it certainly enables delivery to get decent responses to poorly immunogenic antigens.
I think in that last slide you laid out that really nice map of what made the partnership successful. But from just, sometimes it comes even down to the simple things like the logistics. And, you talked about shared students that you've had. Has there been, really concerted effort to making sure that all runs smoothly and is feasible around, accommodation, all that kind of stuff?
Yeah, I think what made the student thing work was both UQ and Monash were really accommodating in that. So Kirsten was formerly enrolled as a UQ student, and when she wandered off down to Monash, she was given the same rights and privileges and access and everything as a Monash student. And likewise when she came back up here. So that reciprocal collaboration across universities, I think, is really important for students, and it just enabled simple things like running across borders to be very easy. The close communication is another big thing. Marie's one of my favourites, and she phones me every 2 minutes, and so that kind of that level of rapport and trust and that close, constant communication keeps bouncing ideas and keeps things moving and keeps momentum going as well. And I think that's really important to have that relationship. Did
you meet through conference networking or through known publications and reach out?
It's been a long-term synergy. So in the Australian DC sort of world, a lot of the really, in the early days, 20 years ago or so, Brisbane was really, really good for human DCs. It ran Jenny, and we had Derek, and I was with Derek, and then we had the Shortman lab and all the people that grew out of that as the mouse DC immunologists. And so we were always around at the same conferences and interacting in a way. And I think it's come from that a lot of sort of complementary expertise and mutual respect for each other that's been able to develop from having the mouse DC biology and the human to be able to synergise to move that forward. So it's it just morphed and evolved, I think. But the real connection started when you know, when we started talking at conferences and they had this cool new molecule and we had these cells that we didn't know what the hell they were and that bound it. So, yeah, and it just morphed from there.
Yeah. Thanks, Mark. Yeah. All right, well, we are over time, so I'll close up there. Thank you very much, Mireille and Kristen, for a really interesting and well cohesive presentation. It was really informative.